Pharmaceutical Sciences. And this event is sponsored by Biju Patnaik University of Technology. So I would like to thank all the officials at BPUT for selecting Roland as a nodal center for this training program. So my sincere thanks to management of RIPS and the coordinator of this program, Biswamohan Sahu, and all the faculty members of RIPS. And my heartfelt thanks to Dr. Kannarao, who readily accepted to enlighten all of us with his knowledge. So I once again welcome all of you and wish this program will be useful for all of us. Thank you very much.
Thank you, sir, for your inspirational words. I am profusely elated to take an opportunity to invite the program coordinator of this online faculty development training program, Dr. Biswamohan Sahu, sir. Sir, please enlighten us with your words of wisdom.
Okay, thank you, ma'am. So due to network connection. So I am very much privileged and feel happy to see the nice strength today's program. So respected principals and other principals of different pharmacy colleges under BPUT, our esteemed resource person, my teacher, Dr. Kannarao sir. So very good afternoon to all of you. So we are ma'am, you can continue. Some network issues there, you can continue next.
Okay, sir. It's a great honor for me to introduce our esteemed resource person, Dr. K.V. Kannarao, sir. Sir had completed his graduation and post-graduation from Andhra University in the year 1991 to 1995. From the year 1995 to 1997, Sir was awarded PhD in Pharmaceutical Analysis from Berhampur University in the year 2003. Sir was faculty in Roland Institute of Pharmaceutical Sciences in various capacities like lecturer, senior lecturer, and reader from the year 1997 to 2004. Sir was research scientist and group leader in Divis Laboratories Limited, Hyderabad. Sir worked as senior scientist from the year 2010 to 2013 and head of analytical research and development nutraceuticals 2013 to 2015 in Laurus Lab Limited, Hyderabad. Sir worked as principal scientist 2015 to 2016 and as an analytical expert from 2016 to till date in Novartis Healthcare Limited, Hyderabad. Sir had presented 4 research papers at various conferences like Indian Pharmaceutical Congress. He has 31 publications in various national and international journals and guided 13 MPharm graduates from their dissertation work. Sir is life member of Indian Institute of Chemists India, Indian Hospital Pharmacist Association, Indian Pharmacist Association, Indian Pharmacy Graduate Association. We are extremely fortunate to have such noble personality with us. Sir, I request you to enlighten the words of wisdom.
Thank you, Aishwarya, for your nice introduction. And I could able to see some of my students as became a faculty members in different colleges, and as well as some old colleagues also. Actually, this is the opportunity to meet all of my colleagues as well as some of the students. That's why whenever I get invitation from Ravi, immediately I used to accept it. So it's my privilege to be a part of any development program which is conducting at Roland Institute of Pharmaceutical Sciences. When I was in faculty there also at that time, I have been part of some training programs at that time. So it's my privilege to meet all of you, and would like to share my knowledge to all of you. So I welcome you all for this presentation session. And shall I start my sharing my screen?
Yes, sir. Yes, sir.
Yeah. I hope you can find my screen now. Yeah. So the purpose of selecting this topic is analytical validations and method transfer strategies. The latest concepts and approaches which industry is following nowadays. Because as in the introductory, we may come across so many theoretical approaches of validations as well as the method transfers, but sometimes in reality it will become difficulty, and we will end up with some method transfers or validations, because the guidelines are different for different countries. So that's why we whenever we are trying to file in a particular country, at that time they used to deny the filing because the validation is not performed as per their country regulations. So for different countries, different as per their regulations, we cannot do method validations, because it is a time consuming process, as well as the approaches are different. That's why as the novartis in line with European pharmaceutical agencies, they come up with some common strategy along with the United States Pharmacopoeia, and trying to do some novel concepts to introduce on the respective chapters of those monographs. Thereby it will be the approaches will be applicable for whole world, not only confined to one or two countries. So the guidelines which currently we have, the slides are not moving. Let me see. Yeah. So first we will see the introduction of what are the concepts and regulations are there. Then the intermediate preparation, reproducibility testing, the new approaches what we follow, and the analytical method life cycle approach, which is the current strategy following following by all the industries, and which will be the part of regulatory requirement in future as well. So as most of us, we know that ICSQ2, which is nothing but validation of analytical procedures, which was implemented and existing since long time. And this is going to be revised in soon to implement the life cycle management concepts. And also USP chapter 1225 and 1226, which describes about validation and verification of compendial procedures. And USP chapter 1092 about the dissolution procedure development validation, and chapter 233 for elemental impurity procedures, and USP chapter 1467, which talks about the residual solvents verification as per compendial procedures, and EP 2.5.12 water, which introduces about semi-micro determination of water content, and ANVISA guidelines, ANVISA RDC 166 by 2017. This is the requirement for Brazil. So any country, if they would like to file in Brazil, they should follow these guidelines. So the analytical validation which we perform, that should also covers all the requirements of this ANVISA guidelines. So this is effective since 2018. And Japan is having its own specific requirements like Kojima design. This is additionally, in addition to our regular validations, we have to perform in order to compile Japanese requirements. And then Chinese pharmacopoeia, chapter 9101. This is a guideline for validation of analytical methods in drug quality specifications. So the transfer of analytical procedures requirements, USP chapter 1224 tells about the transfer of analytical procedures, and ISPE, that is International Pharmaceutical Engineering Good Practices Guide. This is a one general body where the pharmaceutical industries would like to follow the guidelines designed by this ISPE, and WHO guidelines are there, and as well as ANVISA for this method transfers. So the ANVISA guidelines, it is effective since 2018. The separate requirements which ANVISA guidelines requires is we should have pressure, intermediate pressure for LOQ solutions. Actually, the LOQ pressure is not the requirement for other regulatory guidelines, but this ANVISA, more particular about the LOQ intermediate pressure along with the other validation parameters. And also this ANVISA insists about the impurities. The impurities should be spiked at the specification limits, and the pressure also should be proved at that specification levels. So these are the requirements additionally ANVISA is imposing. And the third one is life cycle management of analytical procedures. So why now the guidelines are insisting for this life cycle management? So since the analytical methods will be developed at the R&D sites, and after development, the validation will be performed at R&D site, and the respective test methods, and then methods will be transferred to quality control departments after the completion of the validation. And once the methods have been transferred to the plant, the regular analysis they used to perform. So as the development is being taking place at R&D, but the end users are quality control departments. And the life cycle of each analytical method is very short period at R&D, because during the development, validation, and method transfers, and sometimes initial release of some development batches, and during the stability study only, they would like to see how the method could able to perform for the analysis. But in the plant or quality control department, there are huge number of batches will be analyzed by using these methods, and thereby they are the key persons who would able to challenge sometimes with the method, because they would not like to adopt any thing what other than given in the analytical method. So whatever we provide the method, exactly they will follow, and they will not follow even any additional step like sonication or shaking until and unless the method specifies. So as an R&D scientist, usually what we do after adding solvent to the respective drug substance or drug product, generally we will shake it, or sometimes if it is not dissolved, we will sonicate it for some time until it dissolves. But we use to mix it to write in the analytical method. So if we are not mentioning those steps, so they simply after adding the solvent, if the drug substance or drug product does not dissolve, simply they will keep that solution aside, and they will come up with the method is not suitable to perform that particular analysis. And not only that, as they are coming across several analyses, sometimes they may face some challenges with the method. So that's why this life cycle management comes into the picture to see how the method could be able to perform during its life cycle, since from its development to transfer, and then even after the product is going into the market. Even marketing products also going to be analyzed, and during stability period also the product is going to be analyzed. So is the method is fine during all this life cycle of the drug product, the method is working fine or not. So if any challenges are there, everything will be noted, and this entire process is called life cycle management. So the USP 1220 is insisting on this life cycle management of analytical procedures, starting with the method development and how the procedure is performing, and how it is during its qualification and verification steps as well. And USP, how the results we can able to interpret by without having any challenges with the method. If anything is there, that has to be immediately notified, and the life cycle management, the main concept of the life cycle management is if any small challenge that is facing with the method, as the method is not performing as per the desired or as per the need basis, then immediately it has to be notified. And if it is required, again it triggers about redevelopment of the method, and sometimes we need to again redevelop the new method in order to fulfill the requirements. So that's why nowadays even QBD designs is more renowned concepts were following during the analytical method developments in order to avoid such challenges in future with the developed method. So in the analytical method life cycle, first it will start with analytical target profile, means actually what the aim of the method we are looking. So what type of the technique, and in which matrix you are going to analyze the analyte. So whether it is a drug substance or a drug product or biological product or whatever it may be. So that should be the first our target, and the second target is the business requirement. Yeah. So after developing the method, do we have any requirement in the it will be added any added advantage to our business requirement or not? So once that analytical target profile is designed, so what type of the method, or what, suppose see simply in case of water content, so which method we want to follow, whether Karl Fisher or colorimetric method or whatever, or chromatographic method, what type of it should be HPLC or UPLC method or gas chromatography. So all these things will be designed during this analytical target profile, and once it is done, then we will go the selection of the technique, and then we will work on the method development, and all of you are working on doing the same thing. Okay, so during the method development, the three factors what we have to see is the risk assessment and robustness testing and control strategy. Why these three are very much needs to assess during the initial phase of the method development. So once we developed a method, and the validation is over, and it goes to the plant, and there if the method is not working as per our desired, so again there is a huge impact on the plant. So sometimes the batches should be recalled back once, because the method, because of the wrong method, the testing has been completed, and after the product has been entered into the market, and then it would sometimes there are few instances it has been identified that the method which was followed is not appropriate. So the results which have been based on the results, the batches which have been already released into the batches should be immediately recalled. So all this, in order to avoid such huge impact, we have to do the risk assessment. So risk assessment means here we will see by changing some chromatographic parameters or laboratory variables or method variables. So or any physicochemical characteristics of the analyte, is there any impact on that method? So because as we are producing several different batches of drug substance or different variants of the drug substances based on the need of cost effective synthesis process and other things. So as the route of synthesis varies, the physicochemical properties of the final drug substance will vary. Though the molecule final structure would be the same, but the physicochemical properties will be altered based on these things. So they may impact the performance of the method, which I am going to explain by giving a couple of examples in future slides. And this robustness testing. So in the robustness testing, as you do by changing different columns and different instruments and different grades of solvents, chemicals, and other things, we will do the assessment in the development lab itself. Otherwise, because what are the columns and other things we use may not be the same as in the quality control laboratories or the laboratories which are located at different parts of the world. Suppose it is an Indian company, mostly they may have quality control department and R&D in the same state or in the same country, but some this is mostly happens in the generic companies. Whereas the companies like Novartis and others, all over world, the labs are will be located, and the method should work at any lab or in any country of that, so that irrespective of their global climatic conditions and other things, the method should work. So that's why we will see the control strategy. Suppose some our hello. Yeah, so the room temperature in India usually about say 22 to 30 degrees centigrade in most of the times, whereas some European countries it is only 8 degrees to 16 degrees only the room temperature. So the methods which develop at our climatic conditions may not work well there. So we will during development cycle itself, we will see what will be the impact on the resolution of the impurities when the room temperature or the temperature, column temperature will be altered. Similarly, at the lower temperature, similarly, the room laboratory temperature for in African countries is about 35 degrees. So how the chromatographic conditions will work well. So all these controls in the control strategy we will mention if the temperature is coming back or set at this particular condition, the impurity profile is so and so. So all these how the chromatographic pattern is changing based on the climatic conditions and based on the different grades of chemicals and reagents, different grades of columns. So all these things will be studied, and that will be mentioned in that development report. So that in the control strategy, all these things will be defined. So sometimes some methods are very, very sensitive. They will not work even we will go to the temperature below 2 degrees or 3 degrees. So in such cases, that has to be mentioned. Thereby, it is the receiving unit responsibility to decide whether the method can be helpful for them to analyze that particular targeted analyte or not. So those things will be discussed in the control strategy procedure. So once it is done, then the method validation will be initiated as linearity operation, accuracy, everything, and then intermediate operation followed by reproducibility. So all these things were done. Then we have to do the method transfers to the other laboratories and as well as the quality control departments for routine testing. So in the routine testing, as I previously mentioned, if they are facing any challenges, so those things also will be time to time will be updated. Thereby, in the controlling changes or mechanism, if it is impacting by some of that laboratory environment of the receiving lab is having some impact on while performing the method, those things will be again defined, and all these things will be discussed in this controlled changes procedures. So this is the analytical method life cycle, starting from development to when the method reaches to the plants. So in the life cycle management concepts, usually the advantages with this life cycle management is it gives the improved understanding of the procedure. So one can able to, by seeing this document, one can able to understand how the method works well during its life cycle. Because here I would like to insist, I would like to inform one more thing is, the life cycle of the drug development, usually for innovator molecule, I'm saying, it takes about 10 to 15 years for the development. And once it is after development, the innovator molecule comes into the market, again 15 years will sole exclusivity will be given to that industry. So it is again about 15 years. So 30 years after that, it will be enter into the generic industry, then generic industry people. So the person who is initially involved in the development of that particular analytical procedure, during the initial life cycle of the drug comes into the market itself, he may not be with the same organization. He may leave at any moment. So if the other person will take up that role, and once if you will go through the simple without having this life cycle management concept, one cannot able to understand the challenges what the earlier team has faced and what are the precautions they have taken until and unless we have documented. So this after introducing this life cycle management, one can understand the method, how it is working from since its development to until the analysis of the final product which is in the market. And based on correlation of all these challenges and time to time updating the methods, we can have a robust procedure which can be worked at any lab or any part of the country. So that will be the one of the advantage, and adequate control of sources of variability. So by studying all these challenges, we will have what are the parameters that are impacting the performance of the methods. So thereby we would like to control of those particular parameters, thereby without having any challenge with the method, one can able to perform. See, because why we are so particular is, suppose if we are facing some challenge, what will happen? there we have to open some, suppose because of the poor performance of the method, we may get some OOS result, out of specification result, or out of expectation results we may get, or sometimes if it is a stability analysis, out of trend result we may get because of the poor performance of the method. So if any such thing is observed, again, it is the duty of the scientist to investigate the root cause, why it was the root cause for getting of this OS or OE or some OOT and or some contamination peak or what it may be. So for investigation, again, we may spend about 15 days and maximum 30 days time will be given to investigate and close that particular investigation. So again, the team has to spend a lot of resources for investigation itself. So if we will have such type of the management concept, one can able to immediately identify what could be the root cause and why we are getting this, and immediately they can able to use the control strategy, and without having any difficulty, they could able to perform the analysis by thereby avoid any of these OS, OE or out of trend results during their analysis. And this concept is also helpful for identifying of any adverse trends. Suppose until some life cycle of the column, the resolution, the theoretical plates, everything is will be fine, and after certain hours of the usage of the column, suddenly the resolution may lose, the theoretical plates may lose, and because of that the impurity may merge, it may not resolve from the nearby peak, and it may coalute with some other, and we may get some OS result. So that any trend, so this can be immediately until to wait to know that those adverse trends, immediately we can able to see as the theoretical plates are slowly getting down or the resolution is getting down, we will try to avoid using that column, and we will change the with the new column, thereby it will give some alert to the analyst. So what are the precautions to be taken after usage of this many hours of that column, or so the theoretical plates are coming this much, that means the column in future analysis it may not work well, so try to avoid the column, we will discard that column and we will use a new column like that. Not only column I'm giving, this is one example, so it will be for different things which we are generally used for different kinds of analytical techniques, we use it. So the during as we are performing the linearity, accuracy, everything regularly, but the new concepts which we follow, other than our regular practices only, I'm trying to discuss here, because the regular thing if you will discuss again, it will not be create any interest to you, and it may the session may bore to you as well. So here. So the method design and understanding for doing that intermediate operations and this. So the method variables, actually some method variables, they will be controlled by means of some controlling procedures like the analytical test method. Suppose in the analytical test method, if we say use C18 column of so and so brand, that will be fine, and sometimes so and so are equivalent we will mention. So any brand of that C18 phase stationary phase will work well, but sometimes in some analysis all C18 phases are not equal, as you know, because of the end cap or the amount of carbon loading and several factors. So those things, if you will do it in the development stage itself and evaluate, those things we will define in the document analytical method itself. So thereby the analyst who is going to perform that particular test method, he will confine to use the materials and the equipment or chemicals and reagents what are defined in that respective analytical test method. or sometimes we are having some standard operating procedures. So these are the documents which will allow the control strategy of using the analytical methods. And sometimes the HPLC equipment also will impact our analysis because simply, for example, signal to noise ratio. So that LOQ is nothing but S by N ratio. So we will generally see the PDA detector will give more noise than direct UV detector, because as you know that PDA will works parallelly at multi wavelengths, and it will because of its working procedure, it may create more noise rather than the UV detector, which detects at a single wavelength. So in the HPLC equipment itself, sometimes we will define use Agilent 1290, 1260 or equivalent, or simply use 1290 only, because if 1290 the method may work, but the method may not work for 1260 or 1200 because of the again several reasons of the detector flow cell, the tubings and everything. So all these things it will be defined, and these things will be evaluated during the method recognition, and thereby one will be these things will be fixed in the analytical method, and we will mention those control strategies. And sometimes the experimental variations could able to get because of see a gradient slope due to the instrument. The HPLC equipment, we generally the gradient methods. If the gradient slope, we may say that the same gradient we may run, but all the HPLC instruments may not respond equally because of that slope in the gradient. So those things we can able to very well understand by studying or by performing design of experiments, DOE studies, and those will be discussed in the method under the method robustness of that analytical method validations. So intermediate pressure testing, which is very much essential to understand the analytical uncertainty when the method is going to be used at different laboratories by and different analysts, and using different equipments, using performing the analysis on different days, columns, whatnot. So here, usually intermediate pressure, in generally, we will perform 6 replicate injections for each type of the analysis, and we will perform. So sometimes 2 independent runs we will perform by using 2 different analysts, or in 2 different days we will perform, or by using 2 different equipments, one Agilent and one Waters, or one Shimaju, one, so like 2 different equipments. That type of intermediate pressure generally we do, and by using the column stationary phases, by using different makes of the stationary phases also we do. This is the simple intermediate pressure what generally we do perform. And but the intermediate pressure with extended approach is very much essential in order to compile our method to be acceptable, our validation to be acceptable by any country of the world. So for that, what we do, so 2 operators. So here operators, or we can say 2 associates or 2 analysts, are going to analyze the sample on 4 different days using 2 different kinds of the instruments, and using 2 different columns with 3 sample preparations per day. So usually we do on single sample preparation. The 3 sample preparations concept we introduced it because of the requirement of ANVISA. ANVISA will not accept with a single preparation duplicate determination, always duplicate preparation. Even for linearity also, no other guideline will insist. We generally we do linearity with a single concentration, and from that we will do some dilutions, and with a single solution we will inject it. But this ANVISA to perform linearity as per ANVISA guidelines, we have to prepare replicates of each concentration, and we have to inject from that mean of that replicate injections. We have to plot the linearity. So that's why here itself we are introducing in the intermediate pressure of replications of 3, thereby ANVISA can also it will accept. And also we will combine the method transfer analysis as a part of reproducibility. So reproducibility analysis, suppose we do at R&D, the method validation if we are doing in R&D lab. So during validation, generally what we do, we will write a transfer protocol, analytical method transfer protocol, and it will be sent to the quality control laboratory or the other laboratory where the analysis also will be in future. It will be taking place. So once the method protocol will be reviewed by the receiving unit, they will come up with their objections, if any, or if they are ready to perform the analysis by using that method or not, or are they, the lab is equipped with all the requirements what are given in the electrical method transfer protocol. So once they are accepted, then the same batch what we are going to analyze at R&D lab, some part of the same material will be sent to the receiving unit, and parallelly the operation will be done at R&D lab and as well as at the plant. So it gives the one analyst is doing at a plant and one analyst is doing it at R&D. So it costs 2 different analysts and 2 different labs, as well as 2 different kinds of the instruments and 2 different climatic conditions, climatic zones, or and so whatnot. So it will cover most of the parameters. So that's why we are clubbing the validation as a co-validation. The term it is called. That means the pressure, intermediate pressure, will be performed at receiving unit as well as sending unit. And the other requirement, Kojima, this is the pressure requirement for Japan. So in order to file Japan, we should perform the pressure again as for Kojima. So this Kojima design requirement is, it needs again different operators, again should perform on different instrument column and different days using this. So generally we will how the Kojima design will be used for the intermediate pressure, that I will going to explain in the future slide. So in the Kojima design we will get 12 results, and by comparing these 12 results also the intermediate pressure will be reported. And intermediate pressure sometimes based on the analyzing of the stability samples or during the stability course itself, because on different days the stability samples will be pulled and we will analyze them, and by comparing that data with the initial data, one can able to report the intermediate pressure. So what is the standard exercise generally we follow is we do the develop the method at R&D lab, and the labs at site 1 and suppose site 2, 2 different sites are there. Then analyst 1 will do at site 1, analyst 2 in different. So this what just now I have explained on 2 different sites. And sometimes in the same laboratory, suppose in 2 different labs, 2 different R&D analysts, they do perform the operation by using 2 different. So 2 different labs of the same R&D center it will be done, or 2 different labs of the different locations of the same thing. It will be done, and the method will be sent to production. So where the method will be released to production to analyze for future analysis. So how to combine these intermediate operations for the transfer method? Because in order to again transfer the method at the receiving set, again they need to perform once again. So that's why in order to avoid several analysis, we will combine the co-validation approach here. Again, during the method transfer, the receiving lab, they need not perform again precision. See the intermediate precision data, how it will be evaluated in the transfer analysis. Suppose here the water content method by using Karl-Fisher titration. Suppose at site 1 we do 6 replications for method precision. The results are say under site 1, operator 1 and instrument 1, and site 2 the other part of 6 determinations we get, and we will calculate mean followed by percentage RSD and percentage difference for overall mean and percentage relative difference, and all these things analytical parameters we will evaluate. So intermediate precision, how the data will be evaluated. So we will calculate generally the percentage RSD. So how much amount of the variability can be expected by if the method is performing on two different instruments or equipment or on different days, that can be given when by seeing the pressure or by seeing the overall pressure. So sometimes we used to calculate the t-test and from that also one can able to see the method is performing well or not. So what is the pros and cons of performing the intermediate pressure like this? So here the method transfer requirement for Bezier is already covered under this. So again separate validation or partial validation is not required. And also it will be focus on method variability and not on the batch variability, because we are performing the intermediate precision on two different labs by using the same batch. So we are eliminating the batch variability from one side to another side. So that's why the one variability successfully can able to eliminate the batch variability. So that is one more advantage. What are the cons of this method? So the day-to-day variability in the same laboratory is not addressed here, because in the same laboratory on different day we did not perform. We are directly correlating on one day analysis of the sending unit to the receiving unit. So this day-to-day variability in the same laboratory may also happens. So that data will not be covered under this approach. So if we would like to have it, so that additional experiment we have to perform to prove that day-to-day variability of the variability in the same lab. So intermediate precision testing with minimal approach combinations approach. So in this approach, generally we perform intermediate precision at 4 different laboratories located at different sites by using different operators and by using all different instruments. So we get precision at each site. From that we will calculate percentage RSD and overall percentage RSD variability. Also we can able to get from this data. So this is the design, Kojima design for Japan. To perform intermediate precision as per Japan, the precision we have to design in such a way that as per Japan, that they are very much particular about intraday variability. That's why the precision should be done for 6 days. So here date 1, date 2, 3, 4, 5, 6. So 6 days variability we can able to study. And operator 1 and operator 2, two operators will be there and 2 different instruments. And two different chromatographic conditions will be used, and on each day duplicate analysis results we will able to get it. So for all 6 days, 6 to the 12 determinations will be there, and from that we will conclude the final outcome of this design. So in the operator one, day one, operator one, instrument one, condition one. So here condition one, one can able to have some doubt. What would be the condition? Suppose your mobile phase is isocratic, and it is the ratio of 35 aqueous phase and 65. So in condition one, plus or minus 2% of A and B, we can change it. And condition two, so 35, 65. So in one condition, instead of 35, you change it 33, A and B, 65, it will become 67. And in the second condition, 60, 35 plus 2%, so 37, and there it will be 63, total 100. So like that, that is for the mobile phase. And suppose if the temperature plus or minus 1 degree or 2 degree, that is depending on the control strategy. Suppose your column temperature is 35 degrees. So in condition one, at 33, you can perform, and in condition two at 37. So that alternative condition we can perform. Similarly, so like that in any chromatographic conditions, we will alter and we will perform on different 6 different days by using 2 associates. Thereby we will get the data. So why is the Japan is having this particular Kojima design requirement? Is they are not considering the drug product as a homogeneous. It may include so many excipients along with the drug substance. So at least they need more than 10 preparations. So here 6 days duplicate preparation. So each day 2 preparations we are preparing. So 6 to the 12 preparations we are doing. So minimum 10, at least their preference is greater than or equal to 10 preparations. But in order to avoid the matrix variability in the preparation. That's a some, because of the variability, whether it is really because of the analytical method variability or because of the sample matrix variability. In order to eliminate that, they are very much focus on this matrix variability. And they will also consider the content uniformity results. So in content uniformity, we will take 10 units, 10 tablets or 10 capsules, and each tablet we will inject. So there one can able to see how the content uniformity of that particular dosage form. So this also very much keen for them. The Kojima design has to be done with only one test solution. So this is an alternative procedure, because for intermediate preparation, we are using a chromatographic test method, and each day sometimes we are preparing different test solutions in the case of drug substance. So as the drug substance is same, and if we are preparing on each day, sometimes the test solution preparation may also introduce some variability. So that's why in order to avoid that, Kojima Japan can allow us to use one test solution of drug substance. It is in case of drug substance especially, and can able to perform this intermediate preparation. And for reproducibility, if you perform co-validation, then it will be very much acceptable for as a method transfer activity, and no additional method transfer activities are required if once we do complete this Kojima. So in the Kojima design, after generating the data, see here day one analyst A by using instrument EM and using burette T is getting the value 3.58% of water content, 3.64. Like on 6 different days, in the day 2 analyte 2, second instrument and second burette. And once we go to day 3, same initial analyst is there, but he used a second instrument and used the second burette. And in day 4, second analyst is used whatever the instrument and burette used by the analyst 1 on day 1. And in day 5, the second analyst is going to, the first analyst is going to use the same instrument what he has used on the day 1, but he changed the burette, second burette. And on day 6, second analyst is going to use the same burette what he has used on day 2 is first analysis, and he use the other burette used by the second analyst. So from overall all this we get, so the day-to-day variability about 3.4% variability was observed, and within the day variability it is 2.1%. And method performance by using this method transfer activity as a part of the reproducibility testing. So for reproducibility, we will generate some data at R&D site by using 2 analysts, by using 2 HPLC, each analyst 6 determinations we do. So at site 1, we are having 12 determinations, and site 2 12 determinations. And by comparing the percentage RSD of these 2 sites, we generate the data, and then the method, so the validation done, and the method will be transferred to the production site. This is the reproducibility generally we do. So in order to, if we will perform this co-validation by using the these variabilities, by taking into the consideration and perform the reproducibility, then there is no separate reproducibility to be performed at the other unit, like sending unit 2 analysts will be performed the analysis at the same time. At receiving units also 2 more associates will perform by using another 2 different HPLC columns and 2 different HPLC systems. Thereby at each site 6 determinations will be performed. So 4 6 are 24 determinations at sending unit, and 24 determinations at receiving units. The data will be available. From that we will calculate the percentage RSD, the variability, and thereby we will define how much variability we are getting during the reproducibility of the methods. So the another standard approach generally we follow for reproducibility is we will take one batch at the R&D that is available, and we will perform the reproducibility. And similarly, if in the plant, if already some development batches have been produced at the plant, so they used to take the batch which has been produced at the receiving unit. By using that batch they will perform. So here again there is a chance of introducing the batch variability, but generally standard approach, this thing we will perform, and we at each site 6 sample preparations we do, and sometimes a single preparation with 6 injections we do, and we will compile the data. So by this way we will have some multi-batch comparison data will be there, and sometimes because of the difference in the batch, the results may chance to fail. So once if the results fails means the validation would be impacted, the validation would fail. And similarly, all experiments performed per site are done on the same day. So the day-to-day variation is not captured. So that is the one of the drawback if we will follow the standard approach. The standard deviation on the site might be increased because each site the conditions will be different, so the more RSD value could able to get it. So sometimes the RSD is more, the method may not fulfill the requirement of validation requirement acceptance criteria. So in the co-validation approach, see here in sending unit and receiving unit, they will perform the reproducibility by using the same batch, but on different days, day 1, day 2, day day 3, day 4, and enriching unit 5, 6, 7, 8. So by using different preparations, thereby we are having 48 sample preparations and 48 results. We can have it. So more data we have. So whenever we generate more data, so most of the variability will be nullified, or it will be taken consideration to define the acceptable limits. So it is very easier to compare the within the variability within the lab and between the labs also, and the variability within the intra-day variability or inter-day variability of 2 different locations. So all this data, this much data will be available if we will perform by using this following this co-validation approach. And this is the another example for performing the assay of a particular analysis at 2 different sites. So from this data, as we are having more data, within days variability, day 2 variability, intermediate depression, everything one could able to generate the data. So how this data will be helpful by evaluating the data generated in the during the method precision and the reproducibility. So this data will be helpful to assess the risk to the company, and as well as it will also assess the consumer risk or the patient risk who is going to finally receive the medication. So that risk also it will be it is not at all acceptable. So that will be assessed during the validation of the method itself. So the failure of results means it is the risk to the company, as the validation fails means we cannot perform any method transfers. Then any batch also cannot be released into the market if it does not process the validation criteria. Similarly, suppose as we do not have some intraday variability data and other more data, and with a simple single laboratory data, if we'll perform validation and validation passed, and thereby based on that data batches, sometimes there are some incidences the batches have been released into the market, while the patient during consumption of that medication, it might have been observed at the plant that there is some issue with the method, and I should not reveal one example of our own company. Actually, one product has been released in the market, but during the validation, one placebo peak is not properly assessed during the validation. So as the development team, they know that it is a placebo peak. So during the development and initial their validation, they have not integrated that peak, as they know that it is a placebo. And they have completed validation successfully passed, and batches have been released and product also gone into the market. And all of the sudden, during the stability, some other batch has performed the stability analysis. So they have identified this placebo peak, and it was not reported in the analytical method as a degradation impurity. So immediately they have notified it is a OS. Out of specification, one peak is observed at more than specification limit. So immediately it is notified to the regulatory bodies, and immediately it triggered for a callback of all the product which is there in the market. And fortunately, during the initial investigation itself, it was identified that it is not a degradation impurity, it is a placebo peak. So such things will be risk highly, it impact high risk to the company. That's why during the development and validation itself, each and every observation we have to report in the validation report or control strategy report. Thereby, any other person who is going to use that method, and one can go to the through that document, you can understand what type of the peak it is. So in this case, it is identified as a placebo peak. That is fine. If it is not as a placebo peak, if it is something other, and if that degradation peak is not resolved by that particular method, then the final risk is to the patient, because the patient may consume that medication which is having the degradation impurity more than the acceptable limit. Okay, so these risks can be, this is not at all non-acceptable risk of consumer risk. So that's why all this data evaluation is very, very much helpful or very much needed as a analyst or R&D scientist for us. The statistical significance generally we use based on the data by means of ANOVA, F-test, and t-test.
Vegetables.
There is a huge noise. One can mute. Yeah. So
सारा vegetable का है तू फिर नहीं नहीं खाली vegetable
Ravi, can you warn the person somebody has not? Okay, thank you. So one can able to assess this data by using different kinds of statistical data like F-test, t-test, or ANOVA. So this will help us to tightening the confidence intervals for that respective method, and the lower the number of determinations and the higher the variability generally we observe. So more the data, so if variability is high, automatically the percentage RST will be high. So there is sometimes chance of failure of the data. That's why if you will generate more data, thereby the variability, more data will be considered to fix the limits of that particular parameter, and thereby there is a more likely of passing of the results, and the risk might be reduced by the design of the study, as different kinds of studies what we have gone through the previous slides. So by using different kinds of this statistical evaluation suggested by the regulatory guidelines like WHO, ISP, one can able to demonstrate the significant differences can be acceptable for that respective method, and what is the risk if it is crossing above or beyond the limits that can be known. So to limit the risk to the company, a sufficient number of determinations like 2 analysts per site are suggested by using analyzing 3 different kinds of batches is generally required, and high risk of failure for the study design recommended by WHO as well as ISP, and large resources and efforts are required to reduce the risk, because in order to generate more data, definitely one should use more resources are required to get to generate more data. So it is somewhat the cost of that particular method may go, but in future if you will compare to the callback cost of the to the company and other failures of the batches and other things, initially itself we will pay little efforts, cost and generate more data. It will save most of the time as well as cost to the company. So this is another example for the data generated at 2 different units, and by using t-test toast, the confidence intervals can be minus 0.40 to 1.25% can be acceptable. So any precision beyond this can be acceptable. So like the intervals we can define for that particular methods. So again, this is another example for co-validation. And the data generated by means of reproducibility data and co-validation. Reproducibility testing for method transfer. So for intermediate operation, as if we will do the operation at plant as well as at sending unit, then there is no separate method transfer requirement. So method transfer we can able to avoid. And at each site, the variability, day-to-day variability within day, or overall variability, everything data has been available. The reproducible data has been evaluated and it will be readily available. So the advantages of these new approaches are, so separate method transfer need not perform for Brazil requirement or Japanese requirement. And we will have more data is available, and thereby we can have more knowledge on the method, how it is performing in the due course of variable of several variabilities. And the risk is very, very low by performing the validations and transfers by using these methods. And what are the cons? Definitely the cost will be more as extensive testing is required. And for this, that is Brazil requirement, the LOQ operation is not included here because it is cannot be included in these things. So only that LOQ operation will be done when the method filing is required to do for Brazil. So, and all these designs are not also revealing about the spiking experiments of the impurities at the specification limits. So these are the drawbacks, because if we will introduce them also, again it is becoming more cumbersome to design the experiments. That's why these are not included in the proposed validation parameters, validation designs and method transfer designs. So once the method transfers completes, as we know that the method analysis, the regular batch analysis will be done at quality control laboratories, and the product will go into the market. And during the course, even the batch is released, some amount of that particular batches, those batches will be kept at quality control laboratories, and they will also be studied on a time-to-time basis based on the stability data generated during the development state. Based on that, the shelf life will be assigned for the product. But actually, in the real life cycle, how the product behaves. So to study that one, even the product is released into the market, still the analysis of those batches on to time basis will be done. And if any queries raised by the patients or regulatory authorities or any authorities have any concerns, and if they rise, immediately the batches, some quantity will be available, and immediately it will be helpful for analyzing the batches. So during all these different kinds of analysis, the data is generated, and that data will be continuously monitored, and it will be helpful for the improvement of that particular analytical method, or to define the control strategy of that particular analytical method. And this is also helpful to develop the new method, which can able to consider the critical parameters which have been already evaluated. Because during the life cycle, if it was identified that the method is not actually performing, after some times some degradation impurities are not separating in the you by using the method, then definitely it triggers for a new method development. So at that new development state, so what is the knowledge we have gained during the previous analysis life cycle, that also will be taken and considered, and appropriate development will be method development will be done. And not only that, it also provide the what is the potentiality of your analytical method and how it is the its performance. Do we anticipate any issues with that analytical method or not? So is that will be monitored, and collect and evaluate data related to the method of performance. So on a system suitability, we will define in the analytical method. So we not only consider only the final outcome or final results of the test sample. So we will keep some system suitability parameter. We will define system suitability parameter in the respect analytical method. So even the system suitability data variability also, it will give some idea how the method is performing the due course. So how the retention times are varying, and how the injection precisions are varying, how the resolutions are impacting, how rest-by-N ratios are impacting, all these things will be given. So similarly, replications of samples of standards during the analysis. So it gives the repeatability of the results. Regular analysis of a reference lot or a reference standard, QC check samples at a, so it gives like some intermediate pressure data, and the results of stability studies. We can consider them as an intermediate pressure. The number of recurring deviations or system suitability failures. So not only failure of your result or out of expectation or OS result. Sometimes if the system suitability is also failure, we have to investigate system suitability failure. So if number of system suitability failures are occurring when we are using a particular method, again QA quality assurance department will assign as one action to assess the root cause for these system suitability failures. So if we will having this life cycle management of that method data, one can able to understand, to identify the root cause for these system suitability failures as well. So it will be easy to identify or give justification for the system suitability failures, and the percentage of interrupted analytical sequences. So this is another interesting observation. So during the analysis, suddenly all of the sudden the injection will be interfered. So what we do, we will have say 40-50 injections. So associate at evening time, you will load all the things by seeing initial system suitability criteria. You will see if SST is fail, then you will load all the solutions and you go. But tomorrow morning, once he come and open his sequence, it will be shock for him. The sequence will interfere and it will not complete the complete run. It may stop in middle of the sequence. So if the injection is interfered or interrupted, again we have to investigate that one. Otherwise, QA will not accept the result generated with that. So what is the reason for interruption? So whether it is due because of manual interruption or because of instrument issue. And though if it is manual interruption or because of instrument issue, that is fine. But if it is of your method, so how method will interfere here for interrupting? Suppose what are the solvent system you have chosen in your method. In that particular solvent, if your sample is not completely solubilizing, and if any insoluble particles are there, which we may not identify with our naked eye, but in the micro level, they may clog in the injection needle, or they may clog somewhere, and thereby the injection will stop. So that's why QA will not leave us if we are even SST failures or any injection interruptions are there. So all this data we will generate and we will have, and this also will give how the column is performing. So how many injections the column is working well without having any issues. So that data also can able to know. So analytical procedure lifecycle management, this is the future outlook and the prerequisites what the regulatory bodies are going to be insisted in future. So ICH management committee, it is recommended for revision of ICH Q2, and actually the final decision is due, and it is with focus on sequential approach, including analytical procedure development, component and life cycle management aspects will be introduced in this ICH Q2 R1 as well. And even USP also thinking to revise chapters and trying to include the chapters to address all these things to implement this life cycle management set for different analytical methods. And compilation of these three chapters, 1225, 1226, and 1224, and to a single general chapter, thereby it will be useful to include this analytical procedure lifecycle management as well. And it is also increasing requirements for the pharmaceutical industry. It will help us to gain the continuous knowledge on that particular method throughout the method life cycle. And also it gives more data, there will be more statistical data will be available to evaluate and to see the trend analysis. And also it will help the quality control department to check for their adequate checking purposes during the method performance check as a routine analysis. So this is so before going to this, just a moment, I will share one of my So just please bear with me for a couple of minutes. So this is one of the method methylamine. So this methylamine usually can be determined by GC. Sometimes in the GC method of this particular component, some other peaks are interfering. So they told us it is not possible to develop the GC method. Then as we have developed an analytical method by using the derivation technique. It is derivation, methylamine is derived with N-methyl benzamide. So and we are getting a peak which is chromophoric and can be detected. So in the initial stages of the method, after this method, there are when we inject a sample at high concentration, because the limit is only 32 ppm. So in order to get the LOQ level, the sample concentration has been increased to 10 mg per ml, which is very, very high. So there is a possibility of so many other peaks. So after this benzamide peak, there are some other interference peaks nearby. These are there. So if we are not having some controlling strategy, has not been introduced before studying of our analytical methods in the Novartis. What was happened during the stability analysis and release analysis? Any of this nearby peak of methyl benzamide, sometime it is coming and exactly eluting at the retention time of the methyl benzamide peak. As well, it is a derivative degradation impurity. So until and unless our product is degraded, then only that peak will appear. but the product is not degrading. Because of these interference peaks, sometimes it was reported as a degradation impurity. Fortunately, it is sometimes, most of the times it is LOQ level, and sometimes it is just above the LOQ level also. But actually it is not a degradation impurity of methylamine. So during method transfer, this method has been transferred, and during method transfer, they have faced several challenges, and they're told that this method is not appropriate to perform the analysis at a receiving unit. So then it triggered for development of the new method. So by the time we are already having several data, because we have opened so many OASs out of expectations, and we have investigated, and during those investigations, even some changes in the temperature of the column, it is impacting. Some changes in the mobile phase composition also, it is impacting to move that nearby peaks, and they are eluting at the retention time. So all this knowledge, it will help us to develop a more robust method, and then finally we developed a method, the method wherein we could not find any peak nearer to it. So once after developed this method, it was developed in the year 2018, and validation also we have completed, including Kojima design. We till now we didn't face any challenges. But before that, when we used the previous method, for each and every analysis, we tried to open these investigation actions, and we used to investigate it. Why it is that degradation impurity is forming, whether it is a degradation impurity or not. We used to submit the sample to LCMS, and LCMS, it is not a methylamine peak or methyl benzamide peak, so it is not degraded. So all these things, so much time it saved us, and it is also very much helpful to the company as well, because it will eliminate the investigations. And could you able to see my screen or not?
Yes, sir.
Yes, sir. Yes,
Yes,
sir.
yeah, because the slide is not more, that's why I got it out. So this is with this example we can stop here. So here we are having SF by titration method and SF salt forming agent. So whenever we find the solution like this as an R&D scientist, what we may think the SF by titration means here the reaction takes place between solute and solvent, right? So the solute is the target analyte and solvent is our titrant. If the reaction is taken place, we will get a perfect assay, though the solution is little turbid or whatever it may be. So, but initial development usually we do until and unless we get clear solution, only the method will be developed. Then we have completed the validation. And during method transfer, we got some new variants. So the batch which we used during the method transfer, it is perfectly, it is not perfectly dissolved, but we got some turbid solution like this, but we performed the analysis, and we generated the data. But at plant, they got this turbid solution, and they have stopped it. So the difference between R&D and quality control is whatever you say in the method, that only they perform. They will not perform even single step. So I try to convince them that it is not a problem. You can titrate it, and the reaction is between so and so, and you can proceed. They did not accept. If the solution is not clear, we will not go further for analysis. Similarly, in the case of the second analysis also, both for the same compound in the method transfer, we faced a challenge. Then definitely we can work on this solubility issue, but if you do more, if you will implement more solubility procedure incorporate in the method, again it will trigger for method validation. So because the method validation is already done, and several batches have been released, and stability studies also under progress until 24 or 36 months, the stability studies already completed. So at this moment, if we will completely change the method, it triggers not only for it not only impact the method transfer, method validations we have to do, and all the batches which have been analyzed have been reassessed. They should be reanalyzed, and the stability studies samples also should be reanalyzed. So we have done some brainstorming. So with minimal implementation of some changes in the method, how best we can able to complete these method transfer tasks. Then in the SAB titration method, sonication and grinding step was added. Previously only sonication was there. So because of this sonication, earlier variants are they used to soluble very well. But the future variants, they may be coarser particles than the previous batches. So they're not solubilized. So we have introduced grinding steps. So mortar and pestle, you just triturate it. From that, take the liquids and dissolve in the solvent and sonicate it. Immediately it got dissolved. So because of this grinding step, it is not impact anywhere in the validation. So thereby we could able to evaluate redevelopment or revalidation of the method. Similarly, in the SF salt fuming agent, the procedure is earlier the procedure is stir until the test substance has completely dissolved and titrate. It is the step was given. So until even it is stirred up to more time, it is not getting dissolved. Then that has to be changed like this: stir the test substance for 5 minutes and then sonicate until the test substance. So in the previous case, sonication was not there. That's why they did not sonicate. Even sonication is there. Here in this step, immediately we thought of their sonication was not there. That's why we are end up with this turbidity, and we have sonicated. While sonicated, what was happened? The agglomeration of these test particles have been happened, and more coarser particles we could able to see. Then what we have done, first initially stirring we have done it. In the auto titrator itself, stirring will be there. So stirring 5 minutes we have done it. Then after stirring, the solution became turbid. Then we kept sonication. Then it is a clear solution. Then it is titrated. So by incorporating these slight modifications, so as an R&D analyst, we can implement these without changing the analytical method or something we do, because based on our knowledge. But QC, they won't do like that. That's why. And also another drawback is R&D associates, actually the bench level associate who is performing this analysis. Once he observed this turbidity, he should have immediately notify to his immediate manager. It was not done here because he's an R&D scientist, so he has implemented his knowledge sonication step and got dissolved and is completed. So once it was highlighted at plant, then it is came into the notice into the respect to analytical expert. Then we have acted on it and we have successfully completed. So so this is again triggered. It is almost 3 months delayed to complete the method transfer. If these more data was there and if the method doesn't have shoot up with such an issues, 3 more months early, the product would have been released into the market. So how much impact because of the 3 months delay of the product? So all these things will cost to the company. So these are some of our my knowledge experience. So this is done for my project only. So these are the few knowledge sessions we can have. So our session I have completed from my end, and it's time to address your queries if you are having any. So I could not see actually any queries in the chat box. If anybody can able to read out and inform me also, or you can ask me directly. Hello. Can you hear me? Okay,
Yes, sir.
fine.
Yes,
Thank
sir.
you. So organizers are not available. It seems. So do you have any questions or do you want more information? I'm ready to share you. And if you do not have any queries, you can
Kannada.
reach me. Yes, Ravi.
Hello.
Yes, Ravi.
There one
Dr.
question.
Kannaro,
Yes. Ravi, I can hear you. Yes, Ravi.
yeah, there is a question.
Mhm.
Sunita Patnaik is asking to explain ANOVA.
So this is our statistical performance. So because like a t-test and f-test, generally by generating all this data, we use it to calculate. But see, the statistical formulas and other things we cannot explain here. But the data which can be generated here, and by using the data, if you can go to the any statistical textbook or even our pharmaceutical uh, textbooks also, it will be very well given how to calculate the t-test value, how to calculate f-test, or how to calculate the ANOVA. So here the data, how much is required to in order to perform ANOVA, that has been explained here. So for statistical data, how to calculate and other things, it is a we can discuss in any separate session if you want. How the t-test will be calculated, how the f-test will be calculated on ANOVA, all these things.
Thank you, sir, for your nice presentation. And you have explained detail about the method development and validations in analytical methods. So you have also discussed some drawbacks and some limitations, whatever arrived in the industry also. So it will be very useful for the faculty members who are doing the research work on this area. Thank you, sir, once again for accepting our invitation and giving your valuable time with us. Thank you, sir.
Okay.
Aishwarya ma'am, please proceed.
Thank you so much, Dr. K.V. Kannarao, sir, for such a nice and informative session on analytical validation and method transfer strategy, latest concept and approaches. All of us are going to be highly benefited by going through the facts and figure illustrated by you today. The entire session was too lively, engaging, and educative because of your presence, sir. May I now request our respected principal, Dr. V.V. Ravi Kumar, sir, to felicitate our resource person, Dr. K.V. Kannarao, sir, with an e-certificate of appreciation as a badge of our deepest gratitude and respect from entire pharmacy faculty fraternity. Sir, please.
Thank you, Dr. Kannaro, for accepting our invitation and sparing your valuable time with us. And thanks for the valuable information. And on behalf of all the delegates and Roland Institute of Pharmaceutical Sciences, I would like to present this e-certificate of appreciation. Please accept it, sir.
Thank you very much, Ravi.
Mr. Mohan, sir,
Yeah.
display the certificate. We will mail it to you, sir.
Okay, thank you.
One second, just sharing. due to network, it is taking time.
Okay. Aishwarya Madam.
Yes, sir. Finally, I would like to take this opportunity on behalf of organizing committee reps to place on record our hearty thanks to Honorable Vice Chancellor Sir BPUT, Director Curriculum Development BPUT, Dean Pharmacy, Director and Principal of all BPUT affiliated colleges, above all, resource person of today's event, Dr. K.V. Kanna Rao Sir, for sparing their valuable time for us. We are really enlightened with the knowledge and presence. I would now like to thank our chairperson madam, director madam and principal sir for their enormous cooperation. Special thanks to program coordinator of this event, Dr. Vishwamohan Sahu sir, for timely arrangement and his precise effort for the smooth conduction of the event. I thank everyone for putting effort from planning to the success of the program. Thanks everyone for your presence and for making the event a memorable one. Once again, thank you all.
Thank you very much. Bye.
Thank you, sir.
Bye, Kanna. Now the meeting is ended officially. Sir, Vishwamohan sir.